Clemens, and I'll open the floor for Hans van der Voorn on his contribution as part of the instrumentation area. You have 50 minutes, Hans. The floor is yours.
Thank you. So I'm going to talk about TRPS, which is a system that we've had in place for about 10 years. Single particle measurement, and it's really about confidence. So we had a meeting this morning with a lot of complex instrumentation systems, towers, but I would say generally lacking confidence in the measurement, which holds up progress in nanomedicine in my view. And I've just got some new information on exosome isolation because that's quite exciting, possibly not for everyone here, but I'll put it through. I always start my talks now with the reproducibility crisis, and last year Scott McNeil came up with this data of 450,000 publications, 250,000 in the last 5 years, 80% not reproducible. That's 1,000 papers a week. That is a colossal waste of public money. I made a bad joke at the last talk I gave that DLS is responsible for more unnecessary death and gun crime because it takes so long to get any research done properly with people using DLS. I went back and checked the maths, and I think I was probably underestimating the problem. But yeah, I think that's a real issue that it's time to throw it out and move on. Good measurement, size measurement is essential. We've had this size matters. We've got a number of different reasons for that. It's actually quite easy. Just don't do with DLS. Particle concentration measurement is an essential part of the data set. Not everyone gets that. You talk to nanomedicine people and they treat it like a homogeneous API, but you've got all these discrete packets and you need to understand the nature of the discrete package. You can't just treat it like it's a drug molecule where everything's the same. It's not. So particle concentration measurement matters. Again, not that difficult to get right, but you need to do it. And we've got this thing called high precision single particle charge measurement, a very handy tool. Not everybody needs it, but I'll just show you some applications where that's really useful. And then I'll finish with this what we call high precision exosome isolation, which of course if you don't isolate and manage your sample properly, then measurement doesn't work either. So this is quite an old paper, just quickly showing the differences between the top one. I don't know if the pointer works here. The top one is DLS. So all that they've done is mix 3 monodispersed particle sets. So there's no doubt about what the particles are. Everyone gets it. But you mix them together, measure them DLS, and that's what you get. So that is just so vague, it's useless. If you measure on TRPS, you can actually see the 3 different sizes quite clearly. You also get them in the right proportions, but actually you get the numbers of each as well. So that's an important part of the data set. It's what you actually have and what you need to know. NTA, this is a little bit out of date, but you see this same vague wavy line, but at least it's centered on the particles with the highest number. And the bottom one is a disk centrifuge where you're spinning it out, fractionating a little bit like a field fly fractionation, but you lose a bit of the resolution, and because it's spinning by mass, not volume, you're not getting quite the right answers, and it doesn't do concentration. So of the 4, TRPS gives you all that data relatively quickly, relatively easily, and a fraction of the cost of your field flow fractionation tower. That's about $250,000. This is what the data set should look like. So not like this top thing up here. Where's my pointer working? This DLS wavy, vague wavy line. That's what it should look like. Very precise. It's a histogram. You've got each size bin. This is a trimodal, so you just put 3 different particle types together, and you can see accurately the concentration of each one and in each size bin. So that's how all data, in my opinion, size concentration data should be presented. And as we said this morning, someone presents a Z-average on a DLS and a PDI, it's time to leave the talk. I just give up now. I just think they just don't know what they're doing. Put that data up and think, okay, that means something. How does TRPS work? I'm not going to spend a lot of time on it, but it's an impedance technique. There's a nanopore, a current going through the pore. When a particle travels through the pore, it changes the current, so you get this blockade event here. The depth of the blockade gives you the particle size. It actually gives you the particle volume. You can measure electrophoretic mobility through the width. It's a bit more complicated than that. And the concentration comes from the frequency of the events. And everything is compared with a calibration particle. Why use field fly fractionation? This is just showing what you see on the computer. So this is these are 4 different sizes all flowing through the hole. So each dot represents an event. So you see that going across your screen. So you're fractionating not fractionating the particles, you're fractionating the data. So you can clearly see there's 4 different particle sizes going through. Why have that $250,000 field flow fractionation tower when you can do it for 1/10 of the price? Everything is assured through calibration. The measurement method is actually running calibration particles through the hole, following that with your sample, and the measurement is really every single pulse is just compared with the calibration sample. So everything's always automatically calibrated. TRPS versus NTA, this is not particle metrics, of course, this is the other one. But all this is doing, and I think everybody should be trained to do this in your labs if you've got NTA or DLS. So all that's done here is a bimodal distribution at different ratios. So when you load that up, 75-25, 50-50 on TRPS, but when you put in the NTA, all of a sudden that data starts to disappear. Now, NTA, you've seen some data where they can differentiate a trimodal, but you pretty much have to tell the instrument is a trimodal. Well, that doesn't make much sense. You're supposed to be measuring it from a perspective that you don't know what's there. Concentration now. So the issue with concentration is the field is not used to it. They're used to DLS where you don't have concentration. You just can't do it. So people think you just need a single number. We've moved beyond that because if you have a polydisperse distribution, which you certainly get with EVs, but a lot of nanomedicine products, nanobiologicals, then you're only measuring what you're measuring. And so there are particles down here that you can go from protein size right up to cells. So, 1 nanometer up to 10 microns. So if you just give a single number, it can be quite misleading. So we are now saying you need to specify with your concentration number, your C, the size range. So otherwise it's just a meaningless number, and not many people have got that. But the same applies to your Z average. Your average size is the average size of what you measured, which may not be the average size of what's in the sample. That's an important point. And that applies to every technology, not just to NTA or TRPS. This is just some data showing if you have a very widely dispersed sample, so this was bubbles from 50 nanometers actually up to 10 microns, so you can measure those by doing a series of measurements. This is a little time consuming, but you can get that level of precision if you need to get it. And the concentration of each one varies. So there's just a process about overlapping, making sure you've got sufficient data for each one of those steps. So each one of those colours represents a different measurement on a different nanopore. Now just moving on to charge. So what you measure with zeta potential, everyone gets their numbers. Zeta potential is a function of the particle and the liquid it's in. I mean, that's pretty obvious, but again, not everybody seems to get that. What you measure, whether it's PALS, which is the DLS technique, the zeta sizer technique, they're all measuring electrophoretic mobility. And there's a formula to convert electrophoretic mobility to zeta potential, which I think is fraught with issues, but it's very hard to get people to change. What we measure in a hole is the particle is entering the hole. If you go back to the blockade, there is a, go backwards. There is a full half-width maximum. So the invention that we came up, we realised that the particle enters the hole at this point in the blockade, and the full half-width maximum is the same point in the pore regardless of the size of the particle. So what we do is we time the flow of the particle from that point to the full half-width maximum, and then we vary the pressure, vary the voltage, and out of that you can extract the fluid velocity, which is the convection, the electroosmosis, which is the counterflow of particles against the pore wall, and what's left is electrophoretic mobility. And so then you're doing that particle by particle as it flows through the hole. So this is just some data that was published late last year. So on the left is a series of 5 different particle types. So the yellow colour is where they're all mixed together, and the individual colours are where they're done individually. So they're done individually, and then mix them all together, and then you can see the overlap. And the one on the right is the same particle set done at 2 different settings. So you just to show that it's consistent with the 2 settings. So this is very powerful data that you don't get when you get your average zeta potential, and it can be used in the same way that Clements was talking about the fluorescent technique. This is using charge. So same thing, you're distinguishing particles by their charge, and then if you want to add, instead of an antibody, you can add an aptamer into the mix, and an aptamer is quite highly charged, so there's a number of ways you can do that to tease out what's happening. So there was this morning a talk about ligands, for instance. So there was trying to use field flow fractionation and size measurement for ligand distribution, it's very difficult because the size is changing by 1 or 2%. The charge could be changing by 50%. So it's actually much easier to measure ligand coverage, if they charge particles, with charge than it is with size, because the variable is changing, so your level of precision is much higher. This is some work that was published by a PhD student, Emma Blundell. So she went to a lot of trouble. So the picture, the reason I put up was the picture here. So what that's showing is there are 35 base pair single-stranded DNAs being attached to a particle, and then adding a secondary strand to that. And what she was able to measure is the difference between overlapping at 100% or overlapping at 50-50. It's the same amount of DNA, but she could distinguish the electrophoretic mobility of those two different states, even though they're theoretically the same charge. And the reason is that what you're measuring is a combination of the attractive force to the electrode and the drag coefficient. So it's like having a 400 kilowatt engine in a Ferrari or a 400 kilowatt engine in a four-wheel drive. The Ferrari will go faster. And this is the same. So what you find is that when you start to attach things to nanoparticles, the drag coefficient changes, and this is why this issue of zeta potential is a bit problematic. It's based on a nice round ball, and you start to hang things off the ball, and I think that equation starts to fall down. So there's a technique here where you can measure the formation of particles, and an aptamer is a 3D form of DNA which changes when it binds to a DNA. So you can measure the change in form of that DNA, not just the change in charge. So this is just showing the different rates of flow with a different number of bases. So this is just using DNA as a ligand instead of the carboxylated protein that we saw this morning. This click isn't, sorry. So just a couple of things on particle charge. So what you're measuring electrophoretic mobility. The equation that converts zeta to zeta potential assumes ideal conditions, and I think it's a little dangerous. The drag versus the driving force is what drives electrophoretic mobility. So it's an area of really high interest for research, I think. We can now measure that drag. I think the particle-particle charge is a useful tool in both EVs and nanomedicine. Surface functionalization, protein corona, that sort of thing can be measured accurately. QA, comparative data. Resolution, so what's the resolution of charge? So that paper that was published in December by Robert Vogel and others showed a charge ratio of 44 times 35 base pair DNA molecules on 400 nanometer particles. So he's calculated that on a 100 nanometer particle, that's equivalent to about 15 molecules. And we're doing work at the minute to improve the resolution of charge measurement by 10-fold. So it's not quite there yet, but that's what we expect to come out with in the next 6 months. And then the aim being is that you can detect a single molecule per nanoparticle. So that'll be a single aptamer or a single DNA particle. Just moving on to exosomes. So exosomes are nanobiological particles. So this event is mainly about synthetic particles, but nanobiologicals are a subset of nanomedicine. The difference with liposomes is that exosomes are very heterogeneous. They're dynamic. They're much more complex. They contain very detailed, valuable information as biomarkers, so the states of disease, health, in a multiplicity of ways. They're also proposed as therapeutics. The stem cell-derived exosomes are now taking over a little bit from stem cell therapy, so then you have to measure all that properly. You've got the whole FDA standard issue that you have with liposomes is even more complex with exosomes, and they're now used also as gene delivery vectors. In terms of particle preparation, because this is what we're going to be talking about, there are precipitation kits that have been on the market for a long time. They're now obsolete because they precipitate out everything. Ultracentrification's in wide use, but then you're not getting a pure population of vesicles. Time required, mechanical effect, 120,000 Gs affects the functionality of the EVs. There was a step change in 2014 when, and it wasn't us that invented them, but the introduction of SEC columns gave very pure population, a 15-minute procedure, and then that's now grown to 4 column sizes and 2 gel types, so rapidly taking over from ultracentrification as the better way. It's more efficient, it's quicker, and you maintain the functionality. Now, there's another step change that's coming, so this is just the old style, separation of vesicles and protein. We've now got a 35 nanometer gel, so this is just showing the difference with this limpet 35 nanometer protein with the 35 nanometer gel and the 70 nanometer gel. Challenges are distinguishing the EVs from the LDLs. That's an issue for everybody. dilution and concentration, the system dilutes the sample a little bit, so then you reconcentrate. Measurement of 35 nanometer particles is currently not easily done or not possible, so we're moving to that. By the end of the year, that will be done. And you see these standard NTA profiles where the number of particles drops off dramatically in the smaller size range, but clearly there's a power law in place. So the separation and measurement of 35 nanometer particles will be a good step change for the field. What's the next step change? Is automation. So currently the QEVs are done manually. Now automating it adds a level of precision that we hadn't really thought about when we developed it. So the idea was that it was primarily for convenience. You put the column in place, put your sample in and go away and come back 10 or 15 minutes later and it's all done. So they can be high throughput, so people doing thousands of samples, you can run 5 or 10 in parallel. High repeatability, so it takes away the human error. But we realise this precision and design of fractions is something new. So the idea is that you can, this is the product we're designing and building, that should be out in December. So that's all programmable. The idea is that then the current method of fraction numbers, so we now collect fractions 7, 8 and 9 for instance, that method is obsolete because a fraction is kind of a gross measurement. So what we're now going to be specifying, and we need the whole field to change their view of life, is that you specify the void volume, which is the buffer that flows through the column. You weigh it because you have to measure it, but then it goes to waste. You just ignore it because it's just buffer, it's useless. And then you start a precise volume at 0 at the end of the void volume. So that offers a whole layer of precision that hasn't been around. So you might decide, depending on what you want to do, to go from 0.5 to 2, that would be 0.5 ml to 2 ml post the void fraction. So that exact the terminology is being developed. So we've got the NIH involved, AMC, Mayo Clinic, AMC were the original inventors of the set column, and we've said happy to get comments. But I think that's quite a step change for the field. The columns will be smart columns. Okay, they'll have RFID tags. So this morning Frank was talking about data. So the data will be collected. The fraction columns, they can either have RFIDs or barcodes, but they're not writing on felt pen. So you just record the data into a system. And standardisation is really what this is all about. So it's for nanomedicines as well as EVs. Thank you.
Thank you very much, Hans. And